Hello, everyone. Today I will talk about high-throughput sequencing, which will include the technology of sequencing and their output format, and the very first stage for your all downstreaming analysis, the mapping. First, I want to talk about review about the sequencing technologies. Then I will talk about and here is the figure you already saw, the change of the cost of sequencing of human genome. And the most noticeable point is around 2007, the elbow, that is the coming of next generation sequencing data. And currently, we can even sequencing half billions of 150 base pair paired end reads with a cost of $1,300. That is very amazing. So let's review the history. This is the first generation sequencing called standard sequencing. So we just first split the molecule DNA strands into 4 tubes, and each tube will has all dioxygen triphosphate, which are used to do the to use the reverse coupling property to extend the strand, to synthesize the strand. And each tube will has its own DD-ATP or DD-NTP, which is some modification to the nucleotides, that if a strand or DNA molecule incorporate that DD-NTP, it will stop extension. For example, for tube A, if some of the molecular, when they do the synthesize, it accidentally use a DD-ATP, its extension will end at nucleotide A. So in other words, these 4 tubes will contain various lengths of DNA molecules ending with ACGT. Like in tube A, it will has all the DNAs ending with A, and in the second tube, it will has all the DNAs ending with C. And then we put those running those synthesized DNAs through the gels. Since different DNA molecule with different length, they will run different depth in the gel because their molecule size is different. Then we will see some band on the gel, and the people can read out the original DNA sequence. Like for example, here, the bottom one is corresponds to the shorter DNA molecule, since they are smaller, so they can move further in the gel. So human can then read out after the primer, the first base is C, and then the second base is G from the 4th tube, and so on so forth. And as you can see, this is very low throughput because it relies on human eyes to read that. And here is some screenshot from what we will see from the snapshot of the gel. And here is what we most focus on in this course is the second generation sequencing, which is also called next generation sequencing. And the market of this sequencing technology is dominated by Illumina sequencing technology. So I will explain how they do the sequencing in detail. First, we split our genome into fragments and make that into strands of DNAs. Then we add adapters onto those strands. Then in the Illumina sequencing platform, it will have a huge plate, and each plate will has like a primer of DNA stretched out from the plates. And because of the reverse complementary property, the adapter will attach to the to the plate where it has a corresponding primer stitched out. So that you can see we can fix those DNA strands on the plates. Then this, then if we are doing paired end sequencing, there is one more step. First, we will bend the attached DNA molecule, and the other end will bend down onto the plate, and then we will do the PCR to do a PCR to make the other end also has the same molecular stretch out, and then we cut it from the middle. So for this longer fragment, we have both ends attached onto the on the plates in the sequencing machine. And then currently now each part on the plate will has only one strand of DNA, which is not enough to do the sequencing. So in Illumina, it will like do the clonal do the clone, and then each cell on the plate will has millions on will has very many same DNA molecules stretched out by PCR, and you will see a cluster of the same DNA strand from the same place. And this is just prepared for the sequencing. Yeah.
Is there anything done to ensure that you get complementary adapters on each strand, or are those just
random?
Amplified PCR set? That's how
you mean the this stage?
Yeah, that's set. They each have different adapters on it.
Yeah, this part I think is we will design, like make sure the gray area is near the corresponding black area, so we'll bend down to those. Which part
the question is, if both glass adapters get attached to the same transfer,
I think they would design to avoid that. And here is how the sequencing for one cluster is done. First, the Illumina machine will be flow in all kinds of the nucleotides with some fluorescence molecule attached. So like each A will do some flashing. And also the molecular will also have some specialized, will be changed a bit, so it will extend the cluster base by base. So once it includes that nucleotide, it will stop extend. So for example, so when we sequence here, it will extract the nucleotide C and stops here. And then the machine will wash away all the flowing other nucleotides that are not attached to the cluster. And take a screenshot take an image from the current machine. That is, we will have the color of all the attached nucleotide in the clusters. And then the machine will cleave out the fluorescent part, and also remove the modification of the on the nucleotide. So in the next stage, it can do the extension again. And then the sequencing machine will just do the same operation again and again. So we will extend the template strand base by base. And here is a screenshot of one cycle. This is a screenshot of one cycle, and our cluster is on the top left corner here. So in the first stage, we will see it has a blue color out. So we will know it's that base pair has a nucleotide T. And in the second cycle, we will see a green color there. So that corresponds to a nucleotide G, and so on and so forth. Yes?
So you're getting the order because you're stacking the colors step by step?
Yeah, one base pair each cycle.
How do you ensure you don't have two nucleotides the same color in a row?
So they have the specialized the nucleotide, so it will only extend one base pair in each cycle. So that's why we say cleave the die-end terminating group. So they will attach some terminate group to the nucleotide. So. And the Illumina plate will has like millions such clusters. So that's how it achieves the high throughput. So each cluster will generate one sequencing read. And in recent years, there is also called the third generation sequence data, also called single molecule sequencing data, sequencing technology. Its throughput is much lower, but it generates much longer reads. Like it can, for Illumina sequencing, currently it can generate about 100 to 200 base pair. But for this technology, it can generate megabase reads, which is very useful for genome assembly. But it has a very but its sequencing error rate is much higher than Illumina, and it's also currently under active development. So there are two main companies for this sequencing technology, called PacBio and Oxford Nanopore. So the general idea is that they also have a big plate, and each plate will split into cells, and each cell will contain like a small well or nanopore, and the single molecule will just go through those pores. And in PacBio sequencing technology, it's when it goes through, and it also similar to the Illumina, it will incorporate some fluorescent nucleotide, and some camera will capture the light. And in Oxford Nanopore, when the molecule going through that different nucleotide, when passing, it will has a different electric signal. So that's how it knows which nucleotide from that position is. And there are two YouTube channels, so you can know how they do the sequencing in detail. Any questions?